work that I call it the walking cell, a computer simulation study of metastasis cells. And this code is available in the Zenodo as an open source code. And the idea is to show you, to take the code, and during the presentation, if we have a little bit time, I will show some details of the code. Like the idea is the school is to share and show how to use codes that we have developed. So feel free to take the link and see the code, see the information that are there. Thank you.
Hi. Okay, now it's working. Okay. 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 Okay, let's get started. Hi, everyone. My name is Elder. I'm a researcher in computational biology. I'm a pharmacist, so I hope that I can provide a different kind of view in this topic of protein design for you. I'm leading now a new, recently established group of protein design in NNBU, CNPEM. And today we're going to talk about machine learning for functional protein design. I provide some perspective of protein design field, some historical about this, and something that we are doing now related to that in CNPEM, and try to bring some methods that we are using now, people are using now for protein design using machine learning, especially deep learning. Okay, before I can start, let's just try to make some definitions about protein design. You have two things here. We have what is de novo design and what is protein engineering, right? It's important to make this definition. People use sometimes trying to do their own definition, but this is a common sense, right? So protein design is related to, I mean, you think about a function of a protein, you think about the structure of a protein, and you try to create a sequence that you fold to this structure and perform your function that you need, right? And you do this starting from scratch, right? You are not being based on a natural protein. No, you start from scratch. So this is called de novo design, right? If you have a protein, if you have a natural protein, if you want to optimize this protein, some property of this protein, some function of this protein, you can, for instance, do some mutations. This is called protein engineering, right? For people that are not in the area, just a brief introduction. Proteins are composed by sequence of amino acids. The combination of these amino acids, you generate a structure. We now have good methods for structure prediction. I'll talk about this later. And the structures are commonly associated with a function, right? But here we are most interested in this reverse part here. So we started with a function in a structure. What is the sequence that you can generate to produce this function in structure? Right? This is design of proteins. This is not easy to do because you have like 20 amino acids and you have combined them in the positions of the protein. So, but we also have a structure. So you have backbone atoms and the combination of possible amino acids in each position and the backbones orientations make this problem very complex, right? So usually what people do is to create two steps. So we have an idea, function that you want to, that your protein makes, right? And you generate a backbone. You don't know what is the amino acid in each position of the protein. It's just a backbone, right, in the 3D space. And then, from this backbone, you generate a sequence that you fold to this backbone. But your sequence cannot fold into other alternative states, right, other foldings. You want some sequence that only folds with appropriate energy to this target fold. There are some methods that working by trying to create structures and at the same time create sequence, right? It's called co-design. But usually this is the pipeline that we use for protein design. So you have now sequence, and the end of the pipeline is to validate your sequence, right? You can do this by, for instance, going to the wet lab and perform the experiments to see if your sequence is really folded in your target structure and it's doing what you want. But sometimes you don't have this, so you need to try some in silico validations. So you can use other orthogonal methods to test if your designs, if your sequence are good or not, right? This process involves generative models. And today, we'll discuss more about two of them, which are the diffusion-based models for backbone generation and some graph-based models for the inverse folding problem, right? Has anyone heard about this short story called Ribeiro of Babel? Yeah, nice. So this is considered like analogy because when you are doing protein design, you are actually exploring the space of protein sequence, right? Beyond the natural sequence. In this short story by Jorge Luis Borges, he imagined a library that contains all the books of the world, right? Those books have 410 pages and are created by the combination, random combination of characters, letters, space, commons. If you do this, you can create all the stories that you want, including probably you find this library, a book that is telling the story that living now, our future, everything, right? But it's hard to find a book. If you go to this library, it will be hard to find a book that makes sense to you. You probably even understand what it's contained in the book, right? This is an analogy to the protein sequence space. So suppose you have protein composed by 100 residues. You end up with little higher than 10 to the 130 unique sequence. It's a large space, right? Just as a small portion of this space is composed by natural proteins. Consider like something like 10 to the 20, right? So if we start to combine proteins, for instance, creating chimeras, fused proteins, you start to expand this universe of natural proteins, right? If you do some mutations in this protein, by engineering these proteins, you can also try to expand this space. But you still have a large space to understand, right? So this is what protein design, especially de novo protein design, does. It tries to use methods to understand and try to pick some good sequence from this space, right? It can also help to guide this because it's not, if you go randomly try to find a sequence, probably this sequence will not like fold into a structure that you want, right? So those methods that you talk about, you try to reduce the space and try, no, those 10 sequences are good for what you want. Protein design is not something new. So this is the first case of protein design, designing protein by Bernd Good in 79. He tried to design, actually, he designed 34 residue polypeptide that interact with nucleic acids, right? He did this manually with just experiments. He didn't have like the methods that we have now. At that time, we even didn't have like good structured predictors. So because of this, many people think that it's not possible to design new proteins, right? This is exactly what Peter King mentioned in this commentary here. So pessimists point out that we still could not predict the folding of a protein from its amino acid sequence. But despite this, the pessimists, a few groups set out to design and synthesize a protein de novo. What have we learned from this work? First, the skeptics will now have to admit that it's possible to design proteins de novo, even though we do not yet understand protein folding. He was talking about this study here from William Degrado. In this study, William tried to design 4-helix bundle protein. It's a common fold. We can find this in cytochrome. This kind of fold binds to some small ligands like M group. And his idea was to start with 4 peptides. The peptides individually can fold into a helical structure, right, like this. And they can form tetramers, right? He just changed some residue here to make this core more hydrophobic and increase the stability of the complex, right? And then with this, he just creates some turns between the peptides. So now we don't have 4 peptides, we just have 2. And the last step, he just create a new turn here to connect all the peptides and create a single sequence. Again, this is very manual. But this is what people did in the past. Now, this is a very nice archive about designing proteins available in the PDB, right? What you can see here is an increase. Obviously, we have much more of those proteins, right? But these are just the proteins in the PDB with experimentally solver structures. What you can see here, it's an increase in the number of designs. But you can see another thing here, right? Is the complexity of the designs. So here's just like a peptide. In 2000, people using computational methods create the first protein generated only in the computer, which is called Top7 in David J. Baker and Brian Comey group. And then now we have things like this, like cages for drug delivery. So we can think about protein design in waves. So the first wave is basically doing some manual experiments, trying to change some residues, trying to change some domains. Then with the use of computers and guided by fundamental physical chemical principles, they create a higher success rate for those designs that were expanded using our knowledge about structures in the PDB, using fragment based approach. And now we are living in this era here, the era of AI, because now you have a lot of data in the PDB, and these models are training on those data. sorry, I think it's not good here, but we have now something like a competition between AI companies trying to design the best proteins or trying to produce the best methods for protein design. But my impression is that we still don't have like a protein design method with a perfect success rate, right? So we have many different methods. Actually, every week we see a new method for protein design. But what we need is to create like good pipelines. We need to know those methods like by doing benchmarking and understand how those methods can apply for your case, right? The recent breakthrough success of protein design comes especially from the Nobel Prize from last year of David Baker for computational protein design. But we have other contributions in the Nobel Prize, like Franz Arnold for the direct evolution of enzymes. This is computationally, but this is only experimental. And AI neural networks are also not new in this field. We can see this study from 1904. In this study, they try to create, or actually, yeah, try to create by predicting cleavage sites of signal peptides 1. So basically, you have a sequence, and your objective is trying to predict if this sequence can contain a cleavage site for this enzyme here. We still do this kind of things now, right? So it's nice to see the history about this, right? The method that they use was to generate random sequence and use the neural network as a filter to evaluate the sequence. If the sequence is not good, you generate more. Here's the very basic architecture that they use. So you have like your amino acids with characters, but letters, just amino acid letters, cannot be used directly. So, and also they found that the amino acid sequence per se cannot differentiate between cleavage and a site that is not cleavage by this enzyme. So they decide to create features of those amino acids by just using simply hydrophobicity and volume, right? You can see here. And this directly with the weights goes to an output unit that you predict after activation function if this sequence can be cleaved by the signal peptide or not, right? You saw a lot of maths here, so I'm not going to detail on this, but just since you are talking about neural network architectures, just review some concepts. So a neural network is composed by neurons. We have an input layer that you receive your features. Then this input layer is connected to hidden layers with neurons by weights. And these hidden layers are connected to, can be one or more neurons that you make your prediction, right? You have our prediction here. You have real values, your target here, and you compute a loss by comparing them. And this is a forward pass. You use this as a backward pass by updating your weights to minimize, for instance, a loss function. Inside each neuron, we consider the weights for each. If it's a fully connected neural network, for instance, you consider the weights from all the neurons and add a bias, if it's this case, and input this into activation function. Okay. So just going back to the protein design, why to design new proteins or why to engineering new proteins, right? In terms of protein engineering, we can do a lot of things. You can, for instance, stabilize enzymes. You can create better antibodies. In the side of de novo design, you can create, for instance, new binders from scratch. Those binders can have multiple functions. For instance, bind to viral proteins, work as biosensors, right? So in this field, you have a lot of applications, which is nice. I'll go just through 3 applications, 3 examples, mostly from David Baker Group. This is a very nice example of protein design, design multi-states. So here we have a biosensor. This biosensor is composed by a Luke cage, which is this protein here, and a Luke key, right? These proteins are helical proteins, domains, sorry. And we have, in the Luke cage, you have a cage, you have a latch, right? This latch has this domain here that is called caged SM bit that contains part of the luciferase. Luciferase is the way that you analyze the results, right? It generates bioluminescence. And this part here is the domain that interacts with the target. So this is the Luke cage. The Luke key is basically has another domain, helical domain, with another part of the luciferase. So the idea is, in presence of the target, the system is open, your target is interaction with his domain, and the 2 parts of the luciferase interact together. So now you have a signal, right? This works as a biosensor, but it's nice because you need to design multiple steps here, right? So it's a kind of multi-state design. Going to a totally different application. This is a case of engineering a natural enzyme to make it have another function. In this case, this enzyme is able to degrade an important protein from the gluten, right? And this can be used, for instance, as a therapy for celiac disease. This was created actually in a competition called IGEN. And with this, they created this spin-off from University of Washington. It's called PVP Biologic that were then acquired by Takeda pharmaceutical company. Another totally different application, neurodegenerative disease. So we know that some proteins have the tendency to form fibers because you have, for instance, a specific domain that interact with each other and start to create these repetitions here that form the fiber, right? The idea here is to create de novo protein. You can see that we have a no folding here, but there is a part that is missing, this strand, very strand here, right? This is exactly the location that you interact with the amyloidogenic part of this protein. So when this part here interact with this part is not more available to form the fiber, right? Okay, what was the game changer in protein design the last few years? I believe one important game changer here was the development of structural predictor methods using AI, right? One famous case is the AlphaFold, both versions, AlphaFold 2, AlphaFold 3. I'm not going into detail on this. And how those methods can help in the protein design. They can help by just creating structures, predicting structures. So now you have more data. You add more knowledge also in protein structures, which is good. But they can also help in protein design by generating some confidence scores of the modeling. So when you run AlphaFold, we can extract confidence scores, predicted confidence scores, different confidence scores like PLDTT, PUTM, IPTM, PAE. There are some of those scores that can show, present a confidence of local structures, but there are other confidence scores that can, for instance, predict the confidence in the orientation of different domains, right? So if you have a design, you can try to remodel this design and evaluating the confidence scores, right? Makes sense, right? But we can also use, oh, sorry. Yeah, this is an example of using those confidence scores. We are very interested in design immunological recognition. For instance, design new TCRs. TCRs are receptors of T cells, and they can recognize antigens, for instance, from tumors that are presented by the MHC. This MHC is located in the surface of our cells and antigen-presenting cells. So with this, I'll talk more about this, but if you design new TCRs, you can, for instance, try to guide the activation of the cells to a given tumor, for instance. What we did here was to create a version of AlphaFold for TCR modeling. You want to model the interaction of those receptors, TCRs, with the antigens presented by MHC. So we perform some modifications, especially in the first part of AlphaFold, in the MSA, in the templates, to make this modeling approach better than just using AlphaFold. And it is still better than AlphaFold 3 in most of the case. But the most important thing here is the model confidence. One of the most important things, right? Here's a plot, for instance, when we evaluate the model confidence in the DocQ. The DocQ is basically a measurement of quality of your structure compared with a no structure. If you have a high DocQ, you are more confident that your model is good, right? And you can see that there is a good correlation between the model confidence and DocQ. So if you have, for instance, a new TCR that doesn't have a no structure, if you compute the model confidence when modeling, and if you have, for instance, a model confidence higher, this is a cutoff that you found, but higher than 0.85 or higher than 0.9, you are more confident that this complex can exist, right? But we don't have like just AlphaFold. We have other very important methods like Rosetta-fold, ESM, the different versions of ESM. More recently, Boltz-1, Chai-1. Most of those methods are open source methods that, importantly, also make it AlphaFold open, right? But as I said, those methods are important also in the process of the design. One strategy here is start with, for instance, you have a target function when designing a protein, right? You know a structure that you need. So you can take some sequence, random sequence, for instance, and submit to some modeling like Rosetta, for instance. And you have a prediction, right? You can create some restraints as target functions and go back and try to update your sequence to make the sequence more close to your target structure. We don't need to retrain modeling approach, but you train the relation of sequence. So you are changing sequence and you are seeing the structure. This is different from methods that just use a fixed backbone structure to generate sequence. Because it's possible that when doing this fixed backbone structure, you can obtain a sequence like this one. So you have like two minimums here. This is your target conformation, but there is another conformation that your sequence is also probably, you probably fall to this. So what you want is a sequence that has possibly one minimum that is your target conformation. And it's easier to achieve this by using this approach, which is now also used in a very recently method called Minecraft. Okay, so, but let's go back to our pipeline for protein design. And let's try to talk about each of those parts here. We start by talking about diffusion-based models. The first article that report those the use of those kind of methods was this one from Saul Dixon in 2015. He said that the essential idea of these diffusion models, inspired by non-equilibrium statistical physics, is to systematically and slowly destroy structure in a data distribution through an interactive forward diffusion process. We then learn a reverse diffusion process that restores structure in data in a highly flexible, intractable, generative model of the data. So basically, we have a data, and you destroy this data by doing something. You can, for instance, add noise to this data, and you start with very complex distribution, and just convert this to a very simple distribution, like a Gaussian distribution. And the idea here is to reverse this process, starting from a destroyed data, generate a complex distribution. Yeah, the image is not working, but it's a GIF showing noise to a clean image. But so people start to now implementing the noise diffusion probabilistic model for images. So I think images were the first case of success of diffusion models. So the idea here is to create new images from pure noise. You can think about this as a high-dimensional space of images. You have like 3 clusters of images here. And you want to generate images that are, can be, for instance, sampled from distribution of the capillary images. The way that you have to do this, you can do this, is to start with a random and pure noise image and navigate in this space until you have an image related to this one. But to do this, you'll be like impossible to do this if you don't have like a guide that I show you later. But there is also a very important step here, that is the forward diffusion. This process is the process that guides your training to make the reverse diffusion possible. So, the diffusion process has 2 main parts: the forward diffusion and the reverse diffusion. In the forward diffusion, you start with your image, a clean image, and start to add noise to this image until you have your pure Gaussian image, right? Sorry, we are adding Gaussian noise to this. In the reverse process, you can start with Gaussian image and generate a clean or new image close to or trained the image that you used to train. This is done in multiple steps in which calculate the probability distribution of, for instance, xt conditioned based on xt minus 1, which is the previous step. The same thing here, but in the opposite direction for the reverse diffusion. But these are our concepts. We actually do not code like this. But some other important things here are the schedules. So the schedules determine the amount of noise that you add to your image. You can see here, for instance, that to get this probability distribution of xt conditional to t minus 1, you sample from the distribution of xt minus 1. But if your schedule, the beta t here, is very low, close to 0, you are basically sampling from xt minus 1. But if your beta t is very high, close to 1, this will be close to 0, and you are basically sampling from a random Gaussian distribution. This is not done in a single step, but you can, since you have all the schedules for each time step, you can compute a cumulative product and determine xt conditional directly to x sub 0. So you can control the forward process, right? But the reverse process, the reverse diffusion, is more complicated. It's computational intractable because you don't know the entire distribution of xt sub 0. So to compute the reverse process, from xt, to get xt minus 1 based on xt, you train a model for this. But we are talking about denoising, but the model actually is not denoising an image. The model, you predict the noise of the image. So you have three main steps here. The first step, so the first thing that I need is data for training, right? So you have a lot of images, and in different time steps, what you do is to add noise to those images until you have a pure Gaussian noise image, right? And this is used for the training. In the training, you basically start with your clean image. You define your time step, and for instance, this one here, in your neural network, you predict the amount of noise that you have here. This is what the neural network you do, right? So given an image here in each time step, you predict the noise of this image. But you know the amount of noise that you have in this time step for this image, right? So this is the way that you train the neural network. Because you know the epsilon here, which is the real noise, and here is your noise that you are predicting for this time step. And what you want is to minimize this function here. So you train your model to predict the noise. Usually for images, people start using UNets for this. So once you have your trained model, now it's time to generate your images. This is the sampling process. So you start with this X capital T here, like totally noise image, and you go step by step until you have your, not cleaned, but your new image. For this, if you look at this image here and give to your training model will predict the noise of this image in this time step. But it will be hard. Actually, it will predict the noise based not in the previous time step, but what will be the clean image, right? But it's hard to obtain something good in the first time step, right? So we predict the noise of this image, and then we remove that noise to get your clean image. But since you are in the first step, you are not confident of this. Because of this, you add a bit more noise. And do this step by step until you achieve X sub 0. You can see this in another way. This is a Minus data set from Hugging Face. So you have some input data with numbers. You group the data. Here's an increase of noise from the left to the right. You can see that we don't have much noise here. So if you submit this into the neural network, the prediction is good. But if you take this and submit to our trained neural network, probably you see something that is indistinguishable. I don't know what this is. So because of this, the importance of doing this step by step. So you can think about this like this. So you have these images here, pure noise. You make your predictions. So I think this is correct image based on this noise data. But you are in the first step. You are not confident about this. So you add more noise to this image here, obtain this, and make the prediction again. And you do this over and over until you have your predicted images. This is how diffusion works. There is a lot of variations. But how you can apply this for proteins to generate new backbones? This was one of the first methods that applied diffusion for proteins. Usually, most of the methods that were used for text or images, videos, people start to apply this in the protein field, right? So this is a method called RF diffusion. I'll just give some examples of how it works. So following the same principle of diffusion model, you can have your protein, your backbone, and start to add noise to this backbone until you have like pure noise. You cannot distinguish what is this. Doesn't seem like a protein, right? And you train a model to make the denoising of those coordinates, right? And by doing this, you can generate a new protein structure, a new backbone structure. Let's go see how this can be done. So RF diffusion is, RF means Rosetta fold, and which is method for structural prediction, like AlphaFold. And in RF diffusion, you basically use the Rosetta fold as your neural network. For instance, people use for image UNets. Here we are using Rosetta fold for predict structures, right? It's your neural network. So you are doing the same thing, but we are just changing the players. Instead of UNet, we are using Rutherford here. So I'm just zooming in a step of the reverse process. So if I want to obtain xt, for instance, xt minus 1 from xt, the reverse process, I take the xt, I feed this into RF diffusion. It will make a prediction. In the first step, this will not be good. And then you perform an interpolation between those three structures and add a little more noise, the same thing that I mentioned for the images. And then you have the xt minus 1. There is a different thing here because this x hat sub 0 here is used in the other step together with xt as a self-conditional mechanism. Obviously, the random noise is not done. This step here is not done in the 3D coordinates. They use a local frame, which is rotation equivariant. Right? This is important for our 3D coordinates. The model is training on PDB, and yeah, I think these are the most important parts. So this is just a view of how the method works in the reverse process. So we start with noised structure. In the last time step here, you make the prediction based on these initial coordinates. What should be the structure, the x hat 0 here? Then we interpolate to these two structures, add a little more noise, and get this structure here. And do this over and over until you have your backbone structure generated. This is usually done for multiple applications. You can use this kind of diffusion models, like to generate symmetric oligomers, design a binder, scaffold, a motif. So you have just this part here, which is very important for your structure. And so you just create a larger structure that help to stabilize this motif here. One important question here is, how close are the generated structures by RF diffusion to the structures in the PDB? So here, they generated structures and compare it by using this score, it's called TM score, to the structures, compare with the structures of the PDB. Higher this value, higher is the similarity in terms of structure. Usually, values above 0.6, 0.5 corresponds to a structure that are not correlated. So in terms of, especially in function. What we see is that you can generate structures that doesn't seem like structures from the PDB. So this is very nice. And as example of binders, they try to create binders for main targets. Here's just how the diffusion process can be seen. You have a trajectory. You can, for instance, identify or say, okay, I have a binder, a protein that will bind to this another protein at this site here. So you are conditioning your diffusion for this. There is not just RF diffusion. We have other methods. But all the methods that use the same principle of the diffusion process. What you change is, for instance, the neural network that you are using, or how you be denoising or noising, create noise into your coordinates, right? This is another example, Chroma. It's also a diffusion model. The principle is the same, but the architecture is a little different from RF diffusion. Here are proteins generated conditional to our vocabulary and numbers. Yeah, I think that's it for now. I think you can stop here. And anyone has any questions? Yeah. Sorry? Okay. Yeah, actually, we don't know what are the amino acids here. This is just backbone, right? So you don't know, these are just coordinates of backbones, but you don't know which amino acid is here, right? You are just generating backbones, but you don't know which amino acids are here in these positions. Usually in this pipeline, you start with this backbone and then use another method to predict. This is the one that we talk later in the second part, to predict the amino acid in each position here. But in diffusion, we don't know what is the amino acid that you have. Any other questions? No? Okay. Have our coffee break and go back to continue. The second part, we'll be talking about the graph-based models. O que acontece é que esses modelos, eles são treinados no PDB. Então, cara, o PDB é um sistema que você tem um cristal lá. Então, você não tem garantias. E eu acho que muitas das coisas que podem ocorrer no sentido de output de não funciona, porque a taxa de sucesso ainda desses métodos é muito baixa. Você tem que produzir centenas, milhares de designs para